Hello everybody, welcome back to another review on our lectures that we have in class, and this is going to be our first of Unit 4, so we're going to go ahead and just jump right in. Okay, so just talking about this unit, what I've noticed with my students in particular is that we really are struggling with the AP questions still. Now it's still relatively early in the year, so that's not unexpected, but I do want to start prioritizing it just a little bit more so that we are really ramping up into what next semester is going to look like. So we still have the guided notes in class, but we are not entirely using lecture, which I know some of you guys are a little disappointed about, but using a textbook and learning how to use a textbook is a really important skill for when you go into your next stage of life. This takes a lot more planning on my part, so I understand that when you guys find it a little bit boring, but I promise you that this is a skill that's going to be really helpful when you go into college, especially in biology. Now, I say that especially about this unit because it's so figure focused. We are looking at lots and lots of pathways, signaling pathways, and if we are not able to interpret a figure, almost all of the questions that you're going to see on your AP exam dealing with signaling, especially in the FRQ section, are going to be really, really hard. It's so important that we nail down that skill of how can we interpret a figure, and so that's why we're using the textbook so frequently. And then we're also drawing a lot. This unit deals a lot with mitosis, meiosis, and the cell cycle, and so yeah, we're going to be drawing a lot because that's the easiest way for us to get the information into our brains, right? So for AP biology and really all of biology, learning how to talk about a figure is a really important skill. The first thing that you should do is try to get out of the mindset of trying to figure out the figure. You are not going to know and understand it right away. Instead, just state what you see. And that doesn't necessarily have to start with biological language. As you move through the unit, you should start using the biological language so that you're really starting to integrate that knowledge into really higher thought, right? But really, we're just stating what we see. That might say this blue thing is moving to this, and that's causing this to happen, right? You don't have to be super specific, especially early on. Before we can get started, we really need to start defining these terms, which is the ligand and the receptor. If you had your book, I'd strongly recommend that you read about these two terms. However, for our purposes for this review, I'm just going to let you know that a ligand is just a signaling molecule. It could be a protein, it could be a lipid, something like a steroid hormone, but it's just going to be something that is a signal. It's acting as a signal from the outside environment typically. Now, a receptor is a protein, and this protein is the thing that is going to receive the ligand. And so the ligand binds to the receptor. The receptor is a protein. Now, hopefully you remember how often I say this, structure dictates function. When the ligand binds, it is going to change the shape of the receptor. When it changes the shape, a whole bunch of activities can occur. We're going to see lots of examples of this today and really put it all together. But the idea here is a ligand is a small molecule that is going to bind a protein called a receptor, and that receptor is going to change shape. That's really important. Now, for the rest of this review, it's important because I'm going to be using these terms, ligand and receptor. So, instead of walking you through all the reading, I was going to do that originally. I think that for review purposes, it's more helpful if I just go through the figures with you. So, looking at this figure, this is the first figure that I asked you to talk about with your peer. We have the difference between local signaling and long-distance signaling. So, remember that we are interacting with our environment, and it could be at a very local level or a whole organism level, or potentially in the outside world as well. But if we're dealing with local signaling, it's where we're dealing with adjacent cells. We call this paracrine signaling. And so paracrine signaling is going to be where a secretory vesicle is going to form. It'll fuse with the cell membrane, and then you may remember exocytosis. It is going to send out the signal. In this case, it's we're calling it a local regulator. This is also the ligand. And then a target cell is going to receive that signal, probably on the surface. There's going to be a receptor that receives that ligand and then does an activity in the cell. Now, I don't know what this activity is. It doesn't say it in this figure, but understand that there's going to be some sort of response. That's the whole point, that there's some sort of response here. Now, synaptic signaling is another form of local signaling, but we're really dealing with nervous our neural cells here. And so really we have something called a neurotransmitter that is going to act as our ligand in this case. The mechanism is somewhat similar in that we're typically going to have like a synaptic vesicle form and then release the neurotransmitter. But the key thing here is that you understand that this is for neural cells. Now, long-distance signaling is a little different. We are not acting on adjacent cells. Instead, the ligand, in this case we're talking about hormones, are going to be traveling through the bloodstream to act on a cell really far away within the organism. So it's not adjacent cells, it's something really far away, and it travels through the bloodstream. That's the key part here. So this is the ways that we can deliver signals either in a very local setting or a long-distance setting inside an organism. So before we move on, make sure that you can answer these questions, and then we're going to talk about the next figure. Okay, so the next figure here is dealing with what happens once that ligand actually reaches the target cell. And so we have a signaling molecule, aka the ligand. It's going to bind the receptor. Remember that a receptor is a protein, and because it's a protein, it can easily change shape, and if it changes shape, it'll change function. In this case, the signaling molecule binds the receptor, it changes shape, and is going to be transducing a signal. So it'll typically act on a first molecule here called a relay molecule. Now, what this is going to be dependent on the pathway, so I don't really expect you to know exactly what this is. You'll have to follow a pathway to figure it out. And then this will act on another molecule, and then this will act on another molecule. The key part here is that the receptor is not likely acting on this molecule or this molecule. It's really acting on the first molecule, and then we have this stepwise fashion to get to the end, which would be some sort of cellular response. In this case, it's activation. It doesn't have to be. It typically is, but it does not have to be activation. So this is how we would generally think of a signaling pathway. Now, before we move on, I want you to think about what is the whole point of a signal transduction pathway. It's not so that the ligand and the receptor can just come together and shake hands. There's actually a response that they're trying to form, and that's going to be dependent on the pathway. Now, I asked in class on the whiteboard, think about what these responses could be. I'll give you a couple. It could be to cause some sort of cytoplasmic activity, maybe break down fatty acids, maybe to form fatty acids, maybe to form glycogen. Maybe it's to express some gene. So those are some things, but there's lots and lots of right answers on this. Now, before we move to the next figure, it is so important to me that you understand transcription and translation. So transcription is when we go from DNA to, realistically, this should say mRNA. So DNA to mRNA, and then we take mRNA and we translate it into protein. This is so important. So if I were to ask you what the product of transcription is, you should tell me mRNA or RNA. I would accept that. If I asked you the product of translation, you should tell me protein. Now we have some other terms that we sometimes use for this. Translation, we sometimes refer to it as protein synthesis because guess what? Synthesis means to make. And so translation, protein synthesis is to make a protein. Transcription, sometimes we call it gene expression because your DNA is all your genetic material, gene genetic. And so if I express it, I'm trying to make mRNA. So gene expression is kind of another way to say transcription. Protein synthesis is basically another way to say translation. So the reason why that's important is because we're going to be talking about something called a post-translational modification. Post-translational modification. So that's important for this figure. I would love for you to pause this and then go through it and see if you can figure out what's going on, but I'll go ahead and do it myself. So in this case, we start the same way, right? We have a signaling molecule, aka a ligand, binds to a receptor. This receptor is a protein, which in this case, because the signaling molecule binds, the receptor is going to change shape. If it changes shape, it changes function. And so in this case, the function that it is changing to is to activate a relay molecule. This relay molecule then acts on something called a kinase. A kinase, and I don't have this written on the screen, but I do expect you to know it, so you might want to take notes. A kinase is going to be a protein that phosphorylates something. By phosphorylate, I mean it takes a phosphate from ATP, and it is going to add that phosphate to something else. I'll show you what it looks like in this cascade in just a second. But we have this activated relay molecule that now is going to activate the kinase. What do kinases do? They phosphorylate, meaning they take a phosphate group from ATP and they add it to something else. So this kinase, kinase 1, is going to take a phosphate from ATP, which ultimately converts it to ADP. It takes that phosphate and it adds it to kinase 2. So we can see here we have a phosphate that has been added to kinase 2. This is the only difference between the inactive and the active form. So for most situations, and I'm really emphasizing most, phosphorylation leads to activation. So we take it from inactive. If we add a phosphate group, then we usually activate it. This is not always the case, but generally is. Once again, that phosphate group came from ATP. So we have an active kinase that is going to take an ATP, take the phosphate from an ATP, place it onto a second kinase, and now we have an active kinase. This is kinase 2. Now here's the thing, it's still a kinase. This is a kinase as well, so what do kinases do? They phosphorylate. And so they are going to also take a phosphate from an ATP, and they're going to add it, in this case, to a protein. When it adds it to the protein, it activates that protein. So in this case, we see a phosphorylation cascade where a kinase activates another kinase to ultimately activate a protein that'll elicit a cellular response. Now this is just 2, but we could have, half a dozen kinases in a phosphorylation cascade. So we have simplified it by showing just 2, but it could be many, many, many. Very important. Now you might be asking yourself, well, how do I shut this off? How do I make it go from being active back to inactive? Because I don't want the cellular response forever. That's not good. That would be too much, right? And so if I want to go back, we can do that. It's called dephosphorylation. So we take off the phosphate group using an enzyme called a phosphatase. I'm going to try my best to write that here to the side. A phosphatase dephosphorylates. And so typically for any kinase, there's going to be a phosphatase that is going to undo the work of the kinase. Also, pay attention to the endings of these words. Kinase, phosphatase. You should see ACE there. So what does that tell you? These are enzymes. Kinases and phosphatases are enzymes. So one more time to review, a kinase is going to take a phosphate group from ATP, add that phosphate group to something else. So in this case, we're dealing with another kinase. So kinase 1 takes a phosphate from ATP, adds it to kinase 2, thus activating it. Now kinase, this is also a kinase, and kinases phosphorylate. So we are going to take another ATP, take the phosphate group from that ATP, add it to a protein, activating that protein. If I want to shut off this at any point, I might use a phosphatase to take that phosphate group off. Also, we can have the whole signaling molecule leave. And if the signaling molecule leaves, then everything else shuts off because that receptor changes back to its original shape, thus changing its function. So why do we use this mechanism? And it really has something to do with something called amplification of a signal. So I just want you to understand here that as long as this signal is bound, this receptor is still going to be able to activate the relay molecule. And there might be dozens of duplicates of these relay molecules, maybe hundreds of these relay molecules. And as long as the signal is bound, then this receptor can activate that relay molecule and another version of that relay molecule, another duplicate of that relay molecule, and then we can have another phosphorylation cascade. So we can have just one signal that can potentially activate a phosphorylation cascade, activate this signaling pathway, many, many, many fold. Lots and lots and lots and lots of times, because as long as this is bound, it will just start another signaling pathway over and over again. Okay, So I brought up the idea of transcription and translation, and that it's important to me that you know that even in this unit, because what phosphorylation is a post-translational modification. So let's break down that phrase, right? Post, meaning after. Translation, remember the product of translation is a protein. Modification is a change. So phosphorylation adds a phosphate group to a protein. Well, if it adds a phosphate group, you might be asking yourself, well, why does that phosphate group activate it? In the examples that we've seen, why does a phosphate group activate a protein? And it really comes down, once again, to that idea, structure dictates function. So if I add a phosphate group, well, phosphate groups are, they're relatively small, but they're somewhat bulky, and they are very negatively charged. And so it is very, very likely that if you add a phosphate group, especially in the correct or the right spot, it will induce a change in the shape of that protein. And if you have a change in the shape, it might have been once inactive, but now because you've added something to it, it's going to be active. And so we see that structure dictates function yet again. We are going to see it all throughout this course. You have to believe me. And that's pretty cool. Now there are other post-translational modifications. We focus on phosphorylation, but I want you to understand that there are lots and lots of ways that we can dress up a protein. Think of them as accessories. We can glycosylate them. We can ADP ribosylate them. So there's lots and lots of ways that you can modify a protein. Phosphorylation is just the most common one. Don't forget what kinases do. This figure here is really important for a more specific type of signaling called GPCR signaling. Now, there is one thing that I wish that it was shown here, is that the activated G protein is inactive when it is bound to GDP. So the resting state, meaning the non-activated state here, is a G protein that is bound to GDP. And then, once we have a signaling molecule that binds GPCR, it is going to cause the G protein to change that GDP and exchange it for a GTP. Once it exchanges a GTP, now it's active. And then that can go and work on another enzyme. What that enzyme is depends, once again, on the pathway. But this idea of GPCR signaling is highly conserved. We see it in lots and lots and lots of different systems. We see it in prokaryotes. We see it in eukaryotes. There's lots of fun stuff that comes from this. It means that it probably evolved very early on in evolution and then was co-opted into eukaryotic evolution. So this is really exciting. The idea here is that the G protein coupled receptor, GPCR, sends off this activated G protein to go do work, and then that will potentially lead to another phosphorylation cascade. Depends on what the mechanism is. So it's really important to me that you know that the G protein only becomes active once it binds GTP. If it's bound to GDP, it is not active. And you should also know what GTP is. Well, I'll tell you, it can go GTP and go to GDP, and that should sounds familiar to ATP and ADP, and that's because they're very, very similar molecules. So this is also an energy-rich modified nucleotide. It is guanosine triphosphate, and so it can also be used in a very similar way as ATP. Okay, now this is the more exciting figure. This figure is also GPCR, but it's a little more specific. In this case, they tell you what the signaling molecule is, what the ligand is, and in this case, it is epinephrine, another word for adrenaline. So adrenaline, really it should show it already bound here because the G protein has left, so this is a slightly flawed image. But we have the ligand, aka epinephrine, bind the G protein coupled receptor, which causes the receptor to change shape and causes the G protein to exchange GDP for GTP, thus activating it. So now we have an active G protein that is bound to GTP. Now that it's active, it can go do other work. In this case, it is going to work on an enzyme called adenylyl cyclase. Now, adenylyl cyclase has a very important job. It's a very important enzyme. And I know it's an enzyme because it ends, once again, in ACE. Use that to your advantage. You can see what it's doing. It's taking an ATP, and it is converting it into CAMP. For a second, I want you to ignore the C and just look at AMP. AMP, well, we have ATP, we have ADP, and now we have a third one called AMP, adenosine monophosphate. In this case, it is cyclized, meaning that it forms a ring structure. The structure is pretty fun. You can go look at it, but you will never be tested on that. So what adenylyl cyclase does is it converts ATP into cyclic AMP. Some people shorten it to CAMP. Cyclic AMP is our most famous second messenger. In fact, a lot of times the correct answer, if you ever see cyclic AMP, the correct answer will almost always have second messenger in it. Not always, but almost always. So that's what you should be looking for. So cyclic AMP is a second messenger. Second messengers are small, non-protein messengers. So what they do is, because they're small, they can diffuse really quickly throughout a cell, much faster than maybe another protein can. And so we have this small messenger that can go and act and do work much more quickly. And so CAMP, in this case, acts on a protein called protein kinase A. A very important protein. It shows up in lots and lots of pathways. But protein kinase A is going to signal basically a phosphorylation cascade, because this is a kinase, and remember, kinases phosphorylate. Sorry, I was briefly interrupted, but I think I was leaving off with, since this is a kinases phosphorylate, and so this would probably trigger some sort of phosphorylation cascade to ultimately lead to a cellular response. And so you have an AP Classroom question set on this. The cellular response should make sense, right? If it's epinephrine, you should expect some sort of thing that is going to energize you. And it does. It'll lead to glycolysis, ultimately, at least one of the pathways. Okay. So before we move on, I'd love for you to pause once again and check to make sure that you understand this. But you should know what the role of a secondary messenger is. Remember, they're small, they're diffusible, they move very quickly throughout the cell. How do secondary messengers typically move? Well, through diffusion, and you should know where cyclic AMP comes from. It comes from ATP, which is so exciting. Okay, this next figure here is another mechanism. So we're stepping away from that GPCR, and we're looking at a different mechanism here. In this case, we're looking at ligand-gated ion channels, right here. So in this case, it's pretty easy to follow. The signaling molecule is able to bind. When it binds, it causes the receptor to change shape. And because it changes shape, in this case, it is allowing ions to move into the cell. Now, they're moving here just through diffusion. If this is called a channel, remember, we typically call channels something that is moving through diffusion. And so the ions are just moving through, and then they are going to go do their job. Whatever that signal is, I don't know, but that is what they're going to be doing. So the ligand-gated ion channel allows ions to move in. They will diffuse throughout the cell and then trigger some sort of cellular response. Okay, so we have seen several mechanisms now for how a signaling molecule can get to a receptor, right? We saw paracrine signaling, synaptic signaling, and endocrine signaling. Then we saw the ways that it can get transduced. So the type of reception could be GPCR, and then that could lead to a phosphorylation cascade. But then we haven't really talked about these responses that could happen. And now here it just says activation, but it could be something else. In general, we consider these to be the two main responses. It can either regulate transcription, this is the stuff that I find more fun, or it can regulate activities in the cytoplasm. When we get to Unit 6, we're going to see lots of this stuff, lots of the first one, and we are still talking about some that is regulated in the cytoplasm. Okay, so this figure is really important. So in this case, we are looking at a growth factor as the ligand. Now, typically ligands are pretty well named. So if the ligand here is called a growth factor, that should signal to you that it is probably going to trigger activities in the cell that are going to lead to growth. So that could be cell cycle activation or mitosis. And so in this case, the growth factor binds the receptor. The receptor, of course, is a protein. It'll change shape here, and if it changes shape, it changes function. It'll typically activate a relay molecule, and that relay molecule is going to activate usually a kinase. So it goes from inactive to an activated kinase. That kinase, because what kinases do is they phosphorylate, and so they are going to activate another kinase, and that kinase is also going to phosphorylate because it's a kinase, and it'll activate potentially another kinase, or it could activate something else, but in this case it looks like it's activating a kinase. And so now this activated kinase is physically entering the nucleus where my DNA is. And when it does that, it will find a transcription factor. a transcription factor, just like a growth factor triggers growth in the cell, a transcription factor is a protein that is going to activate transcription. And so this kinase enters the nucleus and it will activate the transcription factor. And I know that it's probably a kinase here because I see a phosphate group on that transcription factor. So this must have been a kinase that enters the nucleus and then activates this transcription factor through phosphorylation. And so you can see then how something from the outside is going to be able to not only regulate activities in the cytoplasm, but also regulate activities in terms of DNA transcription, gene expression. That's pretty cool. The last thing that I want to cover here is one question that is in your AP classroom. And this is actually a really fun question, but it is pretty tough. And so in that, you'll have a figure. The main thing that I want you to think about as we are looking at this is we have an enzyme called cyclic AMP phosphodiesterase that catalyzes the conversion of cyclic AMP to some other molecule. We don't know what that molecule is. It doesn't tell us. Which best predicts the effect of inhibiting this enzyme? So this enzyme converts cyclic AMP to something else. But if we inhibit that, it's not going to convert cyclic AMP anymore. And so this is kind of like almost like two-fold type of question. So you really have to think about it, and I strongly recommend drawing it out. But the way you can think of this is this is acting like as an inhibitor to that pathway for epinephrine signaling. And so if we inhibit the inhibitor, we kind of go back to normal. It's like a double negative. And so the best answer here is going to be C. Because what we had here was it's converting cyclic AMP, which is supposed to activate pKa, to something else. But if we inhibit that process, well, then we now have cyclic AMP again, which can go activate phosphorylase kinase A, or pKa, and that will have it remain active. So I want you to start thinking through these, draw them out as you need, and I hope that your studying is going well. See ya.